researchers, long time no talk. I definitely miss seeing you guys, but I'm excited we're able to keep the research spirit alive, and I'm excited that we're now taking a look at your unknown clones. So I'm hoping that on Monday you were able to complete your post survey and send your email to wssp@waxman.rutgers.edu requesting that they release your unknown sequence. Of course, you only did this if your sequence got sequenced, meaning it was of high quality and your restriction digest and PCR showed lengths over 300 base pairs, right? Because if you remember, that's the indicator that your gene likely codes for a protein. It's not definite, but it helps. If it was only 200 base pairs or 100 base pairs, that indicates that there's not enough nucleotides there to code for enough amino acids to make a functional protein. Of course, this is an overgeneralization because there are functional proteins in your body that are dipeptides. It's just not common. Some of you who did not have great sequences, I do have additional clones and I'll send those out to you today. So what you're going to need to do, unfortunately my screen looks a little different from yours, but when you log onto your DSAP, you'll probably see a little like oval shaped link up in the right hand corner that says add new clones. If you click on that, all of our unknown sequences should be listed there. You'll scroll through and find your clone name. So my name's Jacqueline Katz. If I was looking for my unknown clone, I'd be scrolling through looking for 110JK1.19. Okay. So you're looking for that 110, your initials, 1.19, unless you get an email from me and you're going to be looking for somebody else's clone name, and you'll know who you are. All right, so once you download that clone to your DSAP, it'll show up on your main page. You'll be able to start analyzing it. So once you click on this, this is like the first screen that you see where you need to download your forward sequence. You need to make sure that if you have not done DSAP on your computer before, you have the appropriate software to open your sequence. So for a Mac, that's 4Peaks, and for a Windows, it's Finch TV. If you're a Mac user and you click on this link and it doesn't work, you may need to go to Google and type in 4Peaks sequencing, and it's the first link that opens up on Google. Click on it, get it for free, download it, super easy. Once you have it downloaded, you could download your forward sequence, and for the sake of time, I have done that already, and go to your next page. Hopefully you'll look at the sequence and you'll see nice, clean peaks just like this with high confidence bars, which are these blue bars, which tells me that the sequencing program is fairly certain that right here is a G and right here is a T. Okay, if your whole sequence looks more like, let me see if I can find you a good example. If your whole sequence looks more like this, lots of N's, meaning unknown bases, lots of like triple hump peaks that aren't very clear, lots of overlapping peaks, you might have to answer that your forward sequence is not readable. But I'm hoping for most of us, like in the case of this example clone, it is in fact readable. So now I need to determine the start base and the stop base. So I scroll back over and you have a few options here. So you could just scroll through looking for that ATTA, CGG, CC, and then a run of Gs, which indicates the start of your sequence. You could put it in the search bar and try to find it. Just remember, one of the caveats of that is that if there's a miscalled base, meaning if there's an N in that sequence, it's not going to find it. So I'm going to use the good old scroll method. Here I go. ATTA, CGG, CC, GGGGG. I know my sequence starts right after these Gs. Boom, A59. I type that into my DSAP, A59. Now I go back and I need to find the end of my, I can read, A58. Done. Okay. Now I go back to my A58 and I start selecting until I find the end of my gene, which is in fact the poly A tail. Hopefully it's there. We'll find out. If you do not have a poly A tail, you're just going to select as much sequence as you can until it gets messy and unreadable. The reason you might not have a poly A tail is if the sequencing procedure didn't go all the way through. So as I'm scrolling, I'm like keeping an eye on my peaks and making sure they still look clean, because I do not want to go too far away. Sorry, that looked like a lot of A's, but it's not just A's. So I'm going to keep going. That was a small group of A's, but it's still pretty readable, so I think I'm going to keep going. Oh, there's the shot. These are all my A's. Now I have a few options. Because the signal gets pretty low and there's some ends in there. So what I could do is I could stop before the ends, or it might be my prerogative to continue out until the ends finish and then edit out the ends later. I'm going to stop right before, just preference. And this is A1104. That's my last base, A1104. And then you paste your sequence in here. So Command C, and I paste that in right there. Okay? Then I can move on to the next page, which asks me if I needed to edit my sequence. Here's where I need to flip through and see if there's any of these ends, meaning miscalled bases in here. And the answer for my sequence is yes. So I'm going to hit yes. And then I need to edit this. So it looks like this is right over a red peak. Red indicates T. So I'm going to double click on that, type a lowercase t, and hit enter. Of course, I then need to continue scanning. Here's another one. All right, this looks like it's directly over that big black peak, which is a G. Double click, lowercase g, and I'm on my way, scanning for more ends. My hope is that they don't exist. But of course they might, you never know. And I think for the sake of time and me having looked at this clone before, there are no other ends. So now what I need to do is I need to copy my new edited sequence into DSAP. So of course I need to go back to my very first base, which was A58. And I need to select out all the way to that base 1104, way out here at the end. Let's see how quickly I can get there. Oh, too far. Copy that, and then paste it into my DSAP. Okay? Then comes all our good fun blasting, which I'm hoping you have a fond memory of, using the different databases. And just a quick recap, in case you get started blasting today, your first blast search is against all other published nucleotide sequences, blast N. So these are sequences that do code for a protein. They're sequences that code for tRNA. They're sequences that code for rRNA. There are non-coding sequences. There are promoters, and there's everything in here. So it's kind of like a see what you could see sort of first look. Then you get the BLASTN EST database, which is specifically coding genes, meaning genes that code for a protein. So in this BLAST database, you narrow your search pool, which improves your accuracy. Okay, so we got these two BLAST searches, and then of course you have a BLAST X search, which it's not going to let me go to the next page, but the BLAST X search is going to take your nucleotide sequence, convert it into amino acid sequence, and then search the pool of known protein sequences. All right, so I envision that's possibly as far as you get today. I will be posting another video for Friday's work. And do also remember that you're going to take a look at those two resources about the Lucy project to prepare for our speaker next week. All right, guys. I will be around all day. Shoot me emails if you have problems. I can easily hop on Zoom if you need to share your screen and show me something weird that might be going on with your sequence or your blast. Remember to be patient with blast. Best of luck. This is going to be exciting. Talk to you soon. Bye-bye.